Olga. I'm extremely delighted to be here. Let me quickly start the presentation. I hope you can see my slides. So I'm super happy to give a presentation today and to share the work that I actually still did in the lab of Rudi Ebersold at ETH Zurich. By now, I'm in fact in Munich, the Max Planck Institute for Biochemistry, but the work that I'm talking about today will be focusing on the work that I did in Zurich. And I hope that during this presentation, I can give you some insights and hopefully also some inspiration of how you can expand the concepts of proteomics in general and specifically of DIA to apply them to the analysis of protein complexes and also to assess proteoforms, something that also Lindsay already touched upon in her presentation before. So to introduce the topic of my presentation today, I want to start off with showing this figure which looks at the expansion of molecular diversity in the cell from the genome all the way to the molecular functions in the cell. Because in fact, the genome of the cell only contains in humans roughly 20,000 genes. And this number of genes is then quite dramatically expanded by RNA processing events such as alternative splicing, ending up in tens of thousands of mRNA isoforms, which are then translated to proteins, which we typically analyze by mass spectrometry. So this is actually not the end of diversification, but only the start of more modular and a diverse set of different things that can happen. For example, post-translational modifications, as Lindsay said, can cause phosphorylations or other modifications to the proteins. And proteins can then interact with each other to form protein-protein interactions and also large macromolecular machines, such as the proteasome or ribosome. And as Lindsay already mentioned, the different interactions of the proteins, their localization and their modifications have a very important impact on the protein's function and the different activities they can perform in the cell. So in my presentation, I will now focus mostly on the interactome and how we can extend concepts that you've already heard about for kind of the targeted proteomics to the analysis of protein complexes. And specifically, I will talk about a strategy that we developed in Rudi's lab called SEC-SWOT-MS, which we can use to analyze protein complexes on a global scale and across conditions. And finally, I will change gears a bit and talk about how we can actually get a handle on proteoforms in bottom-up proteomics data and investigate how different proteoforms might have an impact on the protein assembly. So to start off, I'd like to start with a very simple representation of, or like a simple introduction of how protein complexes are commonly analyzed in mass spectrometry. And the gold standard to do this is by affinity purification coupled to mass spectrometry, or short APMS. And what you do in this, in APMS, is you select a specific bait protein and then you fuse an affinity tag to the protein. And then you can lyse the cell and under mild conditions to keep protein complexes intact, and then pull down the bait by the affinity tag. And this will not only give you the bait protein, but it will also pull down all of the interacting proteins. So what this will result in is a very simple interaction network with one central node, which is the bait protein. And you have several interaction partners that are connected to the bait protein via edges. So this is already fairly nice, but one thing that you already can see is the interaction network is still fairly small, and you cannot directly get information about the actual protein complex modules that are there, as for example here indicated by this trimer and the dimer, which are actually separate complexes. So to resolve this, you would actually need to do an orthogonal reciprocal pull down on a second bait, here now indicated with this red protein. And by using the second bait, you can extend, first of all, your interaction network, but you will also already get some insights into modules in the interaction network. If we now, however, want to look at the interactions in an entire cell, then we actually get fairly complex very quickly. So here I'm just showing one example of the massive bioplex effort that has been led by Ed Hudlin et al. And here the goal was actually to get a handle on the entire interactome of the human cell by tagging all human open reading frames. And here you can see the interactome hairball of more than 2,000 baits already. So what I would like to show, what I wanted to show you with this first slide is that affinity purification to APMS actually requires a lot of individual experiments to get a whole interactome. So you can look at APMS as a targeted interactome acquisition strategy. This is similar, for example, compared to proximity labeling, where you fuse the bait protein to an active enzyme that is then labeling proteins that are in close proximity to the bait. And with this, you can get like the surrounding proteins or the microenvironment of a protein in the cell. But both strategies are discrete in a way that the coverage of the complexes that you can investigate with them is directly linked to the number of experiments that you perform. So now the question is if there are any strategies where we can look at the interactome from a more global, untargeted perspective. And the most commonly used approach, and the one that I've also worked with mostly during my PhD, is a strategy called protein cofractionation mass spectrometry, or CoFrac-MS. And here you can take the full native cell and lyse it to keep, again, protein complexes intact. And these protein complexes are then separated by native complex fractionation, for example, by size exclusion chromatography, or short SEC or SEC, or ion exchange chromatography, IEX. The common theme is that you biochemically fractionate the complexes according to some biochemical property, and then you sample a number of fractions across the separation dimension. Each of these sampled fractions is then analyzed separately by bottom-up mass spectrometry, and then what you get are these nice protein intensity chromatograms, where you can see the proteins eluting along the fractionation dimension, and proteins that form a stable complex will nicely co-elute together, as you can see for these two toy complexes here. Now, the benefits of cofractionation MS compared to other strategies such as APMS is that it is actually independent of genetic engineering or the availability of antibodies, and it can, in theory, allow the parallel detection of hundreds of protein complexes at the same time. And so if we go back to the previous slide and we look at the context of or the scope of complexes we can detect in such experiments, we can see that cofractionation MS is not discrete, but the coverage of complexes that you can achieve in an experiment is dependent on whether you do some pre-purification, for example, of specific organelles, such as looking only at the mitochondria or the nuclei. But you can also take the full cell, and you will, in theory, cover the entire interactome of the cell in a single experiment. This is, of course, practically not possible at the moment, but this is mainly due to technical limitations. So we cannot we cannot like cover the entire proteome by our proteomics methods at this stage, but we hope to get there pretty soon. And yeah, so the limitation is not the concept of the data acquisition. On top of co-fractionation MS, there are also other strategies to look at the interactome from a global untargeted perspective. One is, for example, cross-linking of whole cell lysates, which can be done. Another option is to do thermal proteome profiling, or TPP. In TPP, you basically look at protein denaturation curves across increasing temperatures, and what you would expect that proteins that form a stable protein complex actually show very similar denaturation characteristics in this case. And another alternative is to really look at full proteome covariation, where you can leverage very large MS cohorts and look at which proteins actually covary across the entire data set. But in my presentation, I will today mostly focus on co-fractionation.
Isa, there's a question.
Yeah.
How do you handle a large number of baits?
Yep. So this is a very good question, and not necessarily my area of expertise. So I guess so. I don't know exactly what you mean with this question, but you basically have to just acquire data for the baits that you're interested in. So you can, for example, say, okay, you're interested in a certain molecular pathway, and then you select all of the proteins that you know are taking part in that pathway, and you take all of them and analyze them together to get an interactome of this specific pathway or your organelle of interest. But if you have a context or a project like the BioPlex effort, then you have thousands of baits, and this gets very tricky, and you need to have very specific analysis workflows to handle this. I hope this kind of answered the question. So with this, I would now like to move on to analogies between proteomics and interaxomics data acquisition. So if we look at the MS proteomics side and targeted data acquisition, we have strategies such as selected reaction monitoring or parallel reaction monitoring. And what we do in these strategies is we select some peptides prior to data acquisition, and then we specifically target those peptides during our acquisition mode. This is kind of analogous to the situation in APMS, where we, prior to our experiment, select a specific bait protein that we want to investigate in our data acquisition. So on the untargeted data acquisition side, we have on the proteomics side data independent acquisition, DIA, that you've talked about already a lot over the past few days. And here the idea is that you extract peptides from an entire M/Z range and co-fragment the precursors. So what you will see is that you actually have a lot of fragment ions coeluting together, and the situation, and you don't have to select any specific peptides of interest prior to data acquisition. In co-fractionation MS, on the interactomic side, this is actually very similar. So we do the data acquisition for the entire cell together, and you will, in theory, get or like from the conceptual way, you will get protein intensity profiles for all the proteins that you can detect. So if we look at the two strategies on the untargeted data acquisition side, I hope you can appreciate that the data structures actually look very similar. So while on the DIA data set, you have fragment ion intensity profiles across the C18 retention time dimension, and you try to find a peak group of fragment ions that correspond to a single precursor. on the CoFrag-MS side, you have protein intensity chromatograms along this size exclusion chromatography or other fractionation dimension. And here you try to find proteins that coalute and form a protein complex signal.
Isabell, sorry, can I read a question because it might be relevant? Hello,
yeah.
When using SEC or IEX to perform fractionation of native protein complex, how should we evaluate if the complex dissociate during passing of the SEC or IEX columns? So how do you control that dissociation is not happening during separation?
Yeah, that is a very good question. So generally, so our experience is that this actually doesn't happen too often. So what we mainly have, we look at the protein, like the overall protein content that we had, like after the fractionation, we just check the protein content in each of the fractions, and we should see that there are still a lot of protein in the early fractions that are in the high molecular weight range, and these like general total protein profiles, they actually look very similar across like replicate sec fractionations. And if something went wrong during a sample preparation, then you will actually see that your total protein amount in the fractions shifts. And this is a good indication that, that the protein complexes actually fell apart. Okay. So having said that there are actually these conceptual or like similarities between DRA and CoFrag-MS data, I now want to get to the analysis strategies between the different approaches. And in discovery proteomics and DIA analysis, we have on one side the spectrum-centric analysis approach, where we start from the data. And here the idea is that we take the spectra, we perform a library, database search to identify what the data like what signal the data corresponds to us coming from. And a similar approach can also be used for the analysis of large-scale interactome data. And here we start from the data. So here in this representation, each of these little dots correspond to an individual protein. And this matrix shows a correlation matrix between the protein abundance profiles across a co-fractionation experiment. And what you might see is that there are actually clusters of highly correlated proteins. And we can find these clusters and group the proteins to complexes. And then we can check in a database or so whether this complex has been annotated before, and these would be the known complexes. And if they haven't been annotated before, we could identify novel complexes. However, we can also look at the interactome data from a hypothesis-driven direction. And here we can first look again at the peptide-centric analysis approach in DIA, where we start from a spectral library, where we have all the information about our peptides of interest and the fragment ions that we want to look for them. And we then go into the data and try to find evidence for these peptides in our data. So we can use or adopt an analogous approach in the that we call complex-centric for interactome analysis. And here we can take a protein complex database, such as the quorum complex database, and then we can look for evidence of these protein complexes in our data to see if these proteins are actually highly correlated to call this complex detected, or if they are not correlated at all, so this complex would not be detected. So this complex-centric analysis approach is kind of an idea that has been around for some time, and it's not something that we've invented, but it's maybe not always been called like this, but the overall idea has been used in strategies for thermal proteome profiling, where Becher et al. analyzed protein complex variation across different cell cycle stages, and it has also been used in full proteome covariation studies to look at protein complex conservation across humans and mice. And finally, of course, we have in the Eversold lab adopted an automated strategy to look at protein complexes and co-frag-MS data by the complex-centric analysis approach. So we have a question here, I quickly check. So which type of software do you use to assess the statistical significance of interaction results? In other words, to check if the enrichment of a certain protein is of certain proteins is significant. This is a very good question. So usually what we do is we take a protein complex database, as you can see here, and then we also generate a protein complex decoy database. And I will get to this a few slides down the line. But the idea is that we then compare the number of significant, or like the number of detected signals for the target complexes and compare them to the detected signals for the decoy complexes. And then we can build a scoring and an FDR model for this. So now I've basically showed you these two strategies, and I want to give you a bit more perspective of the limitations and the benefits of the discovery and the hypothesis-driven analysis approach in interactomics. So on one side, we have the discovery approach, which has the huge benefit that we can actually determine novel protein interactions, and we don't require any prior knowledge in form of a complex database or so. But it comes at the downside that you might lose sensitivity and selectivity to detect protein complexes because of the large number of proteins that you look at. So on the complex-centric side, we are in a different situation. So we actually require prior information to start our analysis, and we have the downside that we cannot detect novel complexes. However, we have a significantly boosted sensitivity and selectivity because we can use this kind of target decoy approach to really figure out statistically significant protein complex signals in our data. A third approach that is also under the hypothesis-driven strategy is the network-centric idea. And here, instead of using a protein complex database, we can just use a protein-protein interaction, or PPI, network. And here we don't look at an entire complex model at a time, but really just at single protein-protein interactions to see if we find support of this interaction in our data by a positive correlation across our data set. And then we can enhance this edge in the network, or if we don't find evidence, we reduce the weight of this edge to get an updated interaction network. And this approach has actually the benefit that it requires less strict and less well-defined prior knowledge than the complex-centric approach. And it is very scalable across a large number of conditions because not all of the interaction partners always need to be identified in all samples. So this is a fairly new approach that has recently been published by my former colleague, George Rosenberger, in the SECA tool. So one thing that I would like to highlight as well is that the world is not completely black and white, and actually there are also hybrid approaches where you can think of actually combining discovery and hypothesis-driven approaches similar to direct DIA strategies in the analysis of DIA, but I will not go into too much detail about this here. So just as a brief summary of the general concepts of interactome data analysis, I'd like to highlight that they are similar to the targeted and untargeted acquisition strategies in proteomics. These also exist in interactomics, and from the data analysis perspective, untargeted interactome screens have a lot of similarities and similar challenges with DIA data analysis, and the hypothesis or complex-centric interactome analysis strategy can be seen analogous to the peptide-centric DIA analysis, and turned out to be a very powerful strategy that has been successfully applied in different contexts. So there's a question: how many fractions do you generally use? If we have multiple replicates, can we mix the fractions based on Western blot? So generally, so I will get to this, but we used mostly like a lot of fractions, so 60 to 80 fractions by a size exclusion chromatography, which is also suggested. Like I would recommend this if you really want to get a good resolution and you want to look at also at complex intermediates and not only at general broader scales. And the mixing of fractions would probably be possible. We have thought about this, but I think this would make the data analysis way more complicated, and I would not recommend this at this stage, I would say. Okay, so having like discussed now some of the general concepts of interactome data analysis and similarities to proteomics in general, I would now like to focus on the complex-centric proteome profiling strategy by Sex Rothermers that we developed in the Eberside lab during my PhD. And to start off, I would first like to show you the more regularly used discovery workflow for de novo complex identification in CoFrag-MS data, often also referred to as protein correlation profiling. So this is one of these discovery-based analysis strategies where you start from the protein elution profiles, you perform a pairwise scoring of the correlations between the proteins, and then you can use an elaborate machine learning framework to actually group proteins into modules of co-regulated protein complexes. So while this works very well in theory, there are several challenges associated with this analysis strategy. First of all, the situation is not as easy as shown up here, but we actually look at profiles of thousands of proteins at a time. So in our experiments, we had around 5,000 proteins quantified. And due to the limited peak capacity of the chromatographic fractionation strategies that we have, this has the consequence that we are facing a lot of random coelutions of proteins, and this results in a limited selectivity and sensitivity of these de novo complex identification approaches. I will probably quickly look at the questions. So one question is, I would think that all your discovery and hypothesis-driven networks analysis on protein-protein interactions will depend, and that should be validated by other assays methods on the type of buffers. And of course, the number of fractions used. Yes, so this is definitely true. So it generally depends a lot on what kind of fractionation strategy you use, what information you can get. I actually have to check what is the stationary phase in the sec columns, Thomas. I'm sorry. I'm the computational guy in this analysis, but I can send you the reference. So if you check this following paper, you will see all the specifications of the experimental setup that we used. So, oops. How many protein complexes are generally observed in each fraction when you use 60 to 80 fractions? I have to think. So I think this was roughly, so there were quite a lot of complexes. So if we, in a typical experiment, we would find something like around like maybe 400 complexes, and they are not completely homogeneously spread throughout the separation dimension. So on average, I think it was something like, I actually don't, I have to look it up. So maybe it was something like around 10 complexes or so, but might be a bit more. Okay, then there is one question about the machine learning steps for clustering analysis. So basically what is done in these analysis approaches is that you usually take also a prior information from like high confidence protein complexes annotated in quorum, for example, and these are used as like a ground truth data set for the machine learning, and you try to best recover these known complexes, and then based on this train the machine learning algorithm to group proteins according to the results that you would that you basically optimize for these known complexes in the reference database. Okay, so with this, so what I just said is that we have these random coelutions, and we want to increase the sensitivity and selectivity of our analysis. And one way that you can do this, and what has been done by other labs, is to actually use multiple orthogonal fractionation strategies to get rid or like to increase your selectivity. But in the Isabell lab, we had an alternative idea by coming from the DIA analysis. We thought we could use the complex-centric or targeted approach to analyze our data. And so the strategy that we developed is called complex-centric proteome profiling by Swarth MS. And from the technical side, we use size exclusion chromatography at high resolution, sampling between 60 to 80 fractions. And we then perform bottom-up proteomics by DIA or Swarth mass spectrometry to get a highly complete quantitative matrix. And then finally, we play the trick of using the complex-centric analysis strategy, where we use prior information from a complex database to specifically target the complexes in this database in our data. So here I want to again highlight how the complex-centric strategy is an extension of the peptide-centric DIA analysis workflow. So in peptide-centric DIA analysis, we have a spectral library where we have information about specific peptides. Then we go into our highly multiplexed DIA data and extract only the fragment ion chromatograms that we have annotated in our spectral library to do a peak detection to get our peptide signal. In complex-centric proteome profiling, we use prior knowledge from complex databases such as Quorum. We then go into our highly multiplexed SEC profiles and extract only the protein chromatograms of our proteins of interest in the complex that we just query. And then we try to find the protein coelution peak group of the complex in our data. So to perform this analysis, we actually developed the CC Profiler R package, and this is our software for automated complex-centric analysis that allows the parallel and sensitive detection of protein complexes in co-fractionation MS data sets. And as I said, we have a decoy model that can actually perform FDR estimation. So to give you a bit more insight into the workflow of CC Profiler, we take peptide-level protein correlation profiling data, as for example put out by Skyline or Spectronaut, or in our case we use OpenSwarth, and we import these peptides into CC Profiler. Then we perform some sex-specific filtering. And what I mean with this is that we can, for example, filter out peptides that have a lot of missing values, so that don't have consecutive identifications across the fractionation dimension. And then we can also look at sibling peptide correlations, because usually what we would expect if we have a protein without any proteoforms, then all of the peptides from the same protein should have a similar elution profile and they should correlate well. So we can kick out all proteins, all peptides that don't follow these criteria. And then we perform a protein quantification step where we usually sum up the top 3 proteins to get a protein elution profile. Then we can take protein connectivity information and do the complex centric signal detection. And for this, we have 3 main steps. The first is the hypothesis generation. So as I said, you can use protein complexes from complex databases, but you can also use interaction networks derived, for example, from APMS studies such as BioPlex or the string database, and use graph partitioning algorithms such as the cluster 1 algorithm or something similar to group proteins into actual complexes. And then in the second step, we generate a decoy database with a matching decoy for each target. And what we require is that the proteins that are in such a decoy complex are not physically interacting or like grouped together in any of the target complexes. So now we go to the coelution signal detection, and here we implemented sliding window approach, where we go over the fractionation data and try to find regions where our target proteins are highly correlating, and we perform a peak detection. Finally, we can do a grid search across a set of parameters, meaning, for example, the width of the sliding window that is used, or the minimum correlation that our proteins need to have in order to call something a peak group. And then we finally can perform an FDR estimation based on our targets and decoys in the database. And with this, we then get a list of complexes detected in our data. So there's one really great question from Ahmed. If we've tried creating protein profiles from peptides apart from summing them up. So we have tried a lot of different strategies, and I will actually get to this later. But especially if you have different proteoforms, summing up all peptides or like any number of peptides is not the greatest strategy. Other things that we have tried was averaging, but since we filter out, like in this filtering step that I mentioned here, we are actually filtering out any peptides that don't agree with the other peptides. So overall, we should get very homogeneous peptide profiles. And in these cases, the protein quantification doesn't differ so much whether we take the average or the sum or top n, top 1, whatever you choose. Okay, so as I said, we have implemented our analysis strategy in the CC Profiler R package, and we have recently also published a very detailed step-by-step protocol that walks you through the entire wet lab as well as the computational analysis workflow that needs to be performed to get hopefully some nice results for your data. And now I would like to show you some of the insights that you can actually get by applying the complex-centric workflow to your data. And
there is a question. Yeah. So when your output is a continuous area under the curve value for the presence or absence of a peptide, for example, output of skyline, how do you define if the peptide was present or absent in the fraction?
So
Juan, maybe you can ask the question directly.
Yeah.
Hi, yes, sorry, is that the idea, the question came to mind when you were talking about the filtering, that you're checking if, let's say, a peptide was present in consecutive fractions or not. But how would you actually classify if the signal is in there? Like, do you use an intensity threshold for deciding if the area that was integrated is from an actual signal or is just noise being integrated? And if that's the case, how would you define what's the appropriate threshold to use? I'm not sure if I explained myself very well.
Yeah, no, it's a very good question. So generally what we do is we we have we use an FDR threshold on the peptide level. For each fraction separately. So if the peptide drops out of the selected FDR that we choose, then it is we consider this peptide absent from this specific fraction. But this being said, we are actually using a fairly loose peptide level FDR threshold, but rather filter on the protein level. And then we take the sec information as kind of a better way to look at whether the peptide is confidently there or not, because what we expect are these really smooth elution profiles for the peptides, and that the peptides, as I said, correlate well with each other from the same protein. So if this is not the case and the protein has been, for example, quantified by noise in a lot of fractions, then we will filter it out during this filtering step. But yeah, it's a good question.
This FDR level is based on the identifications of the peptide by the database search, or like from an M profit or something like that?
So this is based on something like an M-profit score. So the combined machine learning model of how you score if a signal is present or not.
Okay, thank you.
But it's not a quantification value, but it's the detection value. Thank
you very much.
Okay, so now I get to the actual data. So the first proof of concept study we performed in a human HEC293 cell line, and here we performed Moritz Heusel, who actually generated all of the data, most of the data that I'm showing here today. He fractionated the HEC293 cells into 81 fractions, and each of these fractions were analyzed by DIA swath MS on the TripleTOF 5600 using 120-minute gradients, and this led to the consistent quantification of roughly 5,000 proteins after very strict quality filtering. And using now the complex-centric analysis strategy with quorum as the target hypothesis set, we could confidently detect signals for 572 complexes and at a 5% FDR. And we compared this to a manual annotation benchmark where we manually checked all of the complexes annotated in quorum and manually assessed whether we would call them detectable or not. And with our automated CC profiler workflow, we reached a roughly 90% sensitivity at a 5% FDR. So on top of being able to detect holo complexes, we can actually use the data to also derive information about assembly intermediates. And here what you can see in this figure are the protein subunits of the proteasome. So I'd like to take some time to explain this figure. So on the x-axis, you see the sec fractions from 1 to 81, and they correspond roughly to a molecular weight. And the early sec fractions are according to high molecular weight fractions, because there's a log linear relationship between the molecular weight and the sec fractions, and we can use a calibration standard to match fragments to molecular weights. And the first fractions correspond to high molecular weights, thus large complexes, whereas the higher sec fraction numbers correspond to lower molecular weights for smaller complexes or where the monomers would elute. On the y-axis, you see the protein intensity, and the black vertical lines indicate the complex signals that CC profiler detected. And each of the colored profiles that you see here correspond to one of the proteasome assembly members. And what you can see is that the signal on the left here is the highest molecular weight signal that actually where all of the proteins subunits of the proteasome contribute, and this corresponds to the whole 26S proteasome complex. Here to the right, there's a second peak, which only contains diaphragm beta subunits that form the core 20S proteasome. But what you can also see is that a CC profiler detected several elution peaks in the lower molecular weight range. And we were now curious to see if these might be disassembly products or if they are actually assembly intermediates or something interesting in the proteasome. So what we did is we actually added known assembly chaperones for the proteasome to our analysis, and they are here indicated in red, orange, and black. And what you can see is that these assembly chaperones nicely coallude together with the alpha and beta subunits, and this gave us confidence that these signals actually correspond to an early and late assembly intermediate of the proteasome. And I think this very nicely highlights the modularity and like really the resolution that you can get and into molecular details based on this very global data acquisition approach. So finally, we can also evaluate some global proteome assembly characteristics, because as I said, there is a log linear relationship between the sec fractions and the molecular weight of our of these fractions. And so for each protein where we know the expected monomeric molecular weight, we can assign fractions to being either monomeric or assembled. So when we look at protein signals, we can figure out if detected peak groups are actually in the assembled or in the monomeric range. And we figured out that 66% of the proteins appear in at least one assembled state. And we could further see that a lot of proteins, namely 27%, have multiple peaks along the sec dimension. And I think these two results showed very nicely could give a very nice insight into the diversity of assembly states that are present in a single cell, and that proteins might actually be contributing to alternative functions by being part of different complexes. So while this is, of course, fairly nice to get insights into to proteins in a single state, of course, the question is if we can also look at protein complexes across different conditions. And my former colleague, Moritz Heusel, actually published a very nice study last year where he looked at protein complex rearrangements between HeLa cells in interphase and mitosis. So here I'm just showing a simple example of the chromatin remodeling related BRCA1 complex. And you can see all of the complex members of this BRCA1 complex are eluting here in the early sec fractions in this experiment in interphase. And you can nicely see that all of them are significantly upregulated in mitosis, which is expected due to the experimental setup. So one kind of bottleneck that we then discovered in this analysis is, as I said, we actually used 120-minute gradients in our initial experiment. And what this results to is approximately 1 week of MS measurement time for just a single replicate and condition. And this made this initial comparison between the cell cycle stages extremely expensive in terms of time and, of course, money. So the question was whether we can actually speed up this analysis in any significant ways so that we would make the approach more scalable and actually like a screening technique. So what colleagues of mine did was actually set up a very short EvoSep gradient with only 20 minutes LC gradients. And with this, we can actually analyze one replicate of a sec experiment in a single day. And what you can see in the bottom graphs here is that this shortening of the gradient to 21 minutes results in a decrease in peptide IDs of 30%, and on the protein level, this accords to 20%. But if we look at protein complexes that can be detected, you actually don't lose a lot of information by reducing the chromatographic gradient. And this makes the entire approach much more feasible for larger studies. So what we did was actually use this very short gradient system to investigate the proteome assembly landscape across macrophage differentiation and stimulation. So we took the THP1 cell line and analyzed 3 replicates of the undifferentiated cells. We then differentiated them into macrophages and then stimulated them with LPS. And for each of these different conditions, we extracted the protein complexes and performed the size exclusion chromatography and acquired the data with the 21-minute seq/MS acquisition schemes. So now having this data across a lot of conditions and different replicates, we had to also extend the CC profiler framework. So as before, we have a quantitative peptide matrix and a protein complex annotation as input data. We then import the data, perform a molecular weight calibration, and generate hypotheses. And then we have several pre-processing modules, for example, that include normalization of intensities across conditions and replicates, and an alignment of the profiles between seq runs. We then perform our seq-specific filtering that I mentioned and do protein quantification. Following this initial pre-processing, we can then actually do differential analysis on 3 different levels. The first one is we can look at global assembly state differences. So comparing the assembled mass fraction of a protein. So here we look at a single protein at a time. And again, as I mentioned, we can distinguish the monomeric and the assembled range of the seq fractions. And then we can compare whether there's a shift from the monomeric to the assembled range of the seq range. The second comparison that we can make is we can look at protein peaks without looking at complexes directly. And here we can see that for this protein, we find 3 distinct peaks along the seq dimension. For each of these peaks, we can do a separate comparison to see if, in this case, the monomeric protein abundance decreased during differentiation, which would be the case here. And finally, we can perform the complex-centric analysis, where we look at all the proteins of a specific complex query. And here we detect 3 different subcomplexes of this toy example. And we can, for each of these complex instances, we can do a quantitative comparison. So now I would just want to show briefly what kind of biological mechanisms we could detect in this setup of macrophage differentiation. We could see that generally the proteins that underwent any kind of assembly rearrangements are enriched for terms related to extracellular matrix organization and cell adhesion, which makes sense regarding the morphologic change that the cells undergo during this differentiation process. And upon stimulation, we see that pathways that are related to immune response and to virus infections are actually highly enriched, which is confirming that our experiment should actually have worked. And here I just want to show one example of a protein-centric analysis that revealed the WASH C5 protein that moves from a lower molecular weight peak to a higher molecular weight assembly state. And we can see here that a lot of proteins do such a transformation between different of individual peaks. And this is a specifically interesting protein because this protein we wouldn't have picked up by a standard proteomics workflow because it doesn't change its overall abundance. So the abundance stays actually the same across the 3 conditions, but we see this shift from a lower to a higher molecular weight range. if we look at the complex-centric analysis side, we could see that there were a lot of protein complexes that were actually changing upon differentiation, but only a single complex, the IFIT complex, was upregulated upon stimulation. And you can nicely see this here, that the complex is basically absent in the early two conditions. And here we have a nice high signal of this complex, which is involved in interferon-induced antiviral response. So before I conclude this part on Sex-Rothamus, I would like to highlight one question or answer one question of why we actually want to use DIA for complex-centric proteome profiling. And the simple answer is that we both get more consistent and identification and also quantification. And what you can see here is basically the minimum number of consecutive detections along the sec dimension that we could see in DIA data compared to DDA data acquired in parallel. And what you can see is that generally we have a larger number of consecutive identifications in DIA compared to DDA. Also, the precision of quantification appears to be much better. So what you can see here on the x-axis is the average Pearson correlation of a peptide with its sibling peptides originating from the same protein. And what you can see is that the average correlation for DIA was much higher than for DDA. And finally, these two aspects put together actually resulted in a much better performance in the complex detection for DIA compared to the DDA workflow. However, I have to say that this is all data on the SciEx triple TOF instruments, so this might look slightly different for different instrument platforms. So to summarize this part of my presentation, I hope that I could convince you that complex-centric proteome profiling is able to detect and quantify hundreds of protein complexes in a single go. And it also enables the investigation of subcomplexes and assembly intermediates, and to look at global assembly characteristics, such as changes in the assembled mass fraction. Furthermore, we can look at complexes differentially and look at rearrangements across conditions. And by using very short gradients, this allows us to measure a single experiment per day, making this much more scalable. And finally, I'd like to say, as mentioned before, that DIA really has some impact on the quality of the data that we can get. I quickly look at the question here. If one does not have the possibility to DIA, is it fine to do 30-minute DDA measurements? So there have a lot of studies have been done with DDA, and I think this is fine. I mean, as I said, there are challenges associated with it, and you might not get like the quality of your chromatograms might not be as nice, and the scoring might suffer from it. So yeah, I would generally recommend DIA if you do DDA. I would definitely go for longer gradients than 30 minutes, just to make sure that you actually cover enough proteins. So one other option is that, so I am usually with DIA starting from peptide level information, but you can also use CC profiler beginning from proteins directly. So this might be more optimal for using DDA, because you kind of get rid of problems of single peptides being missing in some samples. And by starting from the proteins directly, this could be easier. Another question by Charlie: can SEC really isolate more than 10,000 kilodalton of protein complex? Yeah, this is a good question. So if we go here, so there is not a high resolution at this really high molecular weights. What we usually see is that in the very early fractions, there are lots of proteins eluting, and they might be membrane associated or like specific to smaller organelles, for example, the lysosomes. And so a really good resolution you will get only in the middle part. So yeah, the high molecular weight range is more tricky. Another question by Thomas, do you think this workflow would be amenable to a middle down proteomics workflow? So this would be fantastic. I would really love to see this combined with middle down proteomics or even top down. I think this would be fantastic. So I think I don't see why this shouldn't be possible to combine. Only problem might be the sample amount that you need. So you need to make sure that you, I don't know what is currently required for having a good middle down proteomics result. Okay, I think these are the questions. So, so with this, I now like to go back to my introductory slide and where I was talking about the molecular diversification in the cell. And I will now change gear a bit and focus on how different proteoforms, for example, derived by alternative splicing or post-translational modifications, can have an impact on protein complex formation by first looking at how we can actually measure proteoforms in bottom-up proteomics. So for this, I want to go back to the general principles of bottom-up proteomics and highlight its limitations. So in a standard workflow, we start from proteins, we digest them into smaller MS accessible peptides, and then we perform our mass spec analysis. And from this data that we generated, we have a computational workflow first back to the peptides by database search or our DRA analysis, and followed by a protein inference step where we map peptides back to the parental proteins. So while this is fairly simple, if you imagine that each protein would have a unique set of peptides, but in the real world proteome, this is much more complicated. First of all, peptides might be shared between proteins of different genes. And this is a fairly common problem, and it's usually tackled by protein grouping strategies where you, for example, assign a shared peptide to the protein which has more unique peptide evidence. But the situation gets even more complicated if we consider alternative sequences derived, for example, by alternative splicing, and even more when we look at PTMs. And in these cases, we would actually have to perform a proteoform grouping to look at an individual functional species. And usually this is not being done in bottom-up proteomics due to these challenges of having disturbed this direct association between the proteoform and the actually measured peptides. And this is done more typically by top-down. So before we go into more details, I want to briefly define what I mean with a proteoform. So proteoforms, in the way that I define them here, are protein variants that originate from the same gene, but that have a unique amino acid sequence and post-translational modifications. And so as I said, typically we do not look at these proteoforms in bottom-up proteomics. However, now the question is there anything we can do to change this? And the answer is yes, because we can play a trick and leverage information across a large number of samples. So in bottom-up proteomics, we have the benefit that it's fairly easy to generate large data sets, and we can use information from sibling peptides across large data sets to evaluate or to find groups of correlated peptides. So here I want to just show one example where we have two toy conditions, A and B, measured in 6 biological replicates. And what you see are peptide intensities that are all belonging to the same protein, or like the same original gene. And what you see is that the majority of peptides indicated in blue follow the same quantitative pattern, and they increase in abundance in condition B. However, there's a second set of peptides indicated in orange that actually decrease in abundance in condition B. And this can be explained by different strategies, but one thing that you can appreciate already is that if you do a classical bottom-up proteomics workflow and you just do protein quantification here, and you would consider all peptides, this would actually result in a fairly bad quantification based on these orange peptides. So now you can tackle these kind of problematic proteins by filtering out the outlier peptides to improve protein quantification. However, the other approach is to actually assume that these peptides might be of biological relevance, and you can try to find individual peptides or groups of peptides that follow a different pattern and try to see if they actually form an interesting proteoform group. So now I will present one a tool that we developed in the Ebersold and Rist lab that is called correlation-based proteoform assignment, or short COPF. And here the idea is exactly that if you have a single protein that is generated from a gene, then we perform bottom-up proteomic analysis. Then all of the peptides in theory should follow the same quantitative trend across a large data set. However, if we have a gene that actually results in different alternatively spliced mRNA isoforms that are then translated to different proteoforms, they can actually no longer be separated in a single bottom-up proteomics experiment. If these two proteoforms are differentially abundant across the conditions that we look at, we can actually figure out groups of co-regulated peptides which will distinguish the two proteoforms. So now we can actually do this automatically by looking at the pairwise peptide correlations. So here on the left-hand side, if all of the peptides follow the same pattern, they all have a very high correlation and you get a very homogeneous pairwise correlation matrix. However, if you have two alternatively regulated proteoforms, peptides of the same proteoform group will have a high correlation, whereas peptides across the two groups will have a low correlation. So the idea in COPF is that we take these peptide correlations and perform a hierarchical clustering and group the peptides into two clusters based on the correlation distance. And then we can calculate a proteoform score based on the within versus across cluster correlation, and actually also calculate an associated p-value to this proteoform score. And what we will get from this is something that looks like a one-sided pseudo-volcano, where we have the proteoform score on the x-axis and log-negative adjusted p-value on the y-axis. And you will see that proteins with multiple distinct proteoforms end up with a high proteoform score and low p-value. I quickly answer some questions. So does COPF apply to DDA or just DIA? So I have analyzed only DIA data with COPF so far, mainly because it strongly benefits from very good quantitative peptide level information. And this is usually not as good in DDA. But in principle, the method is agnostic to how you acquired your data. Then is the COPF applicable to measure PTM or phosphorylation stoichiometries? In principle, yes. So you can measure PTMs, and you can also try to figure out the stoichiometry, or like the occupancies of the PTMs. Although I have to say that I haven't done it myself so far, but it's definitely possible.
Can I add a little bit to that question? Because yes, as it is the case with many PTMs, unless you perform enrichment, you will usually not see them. And that changes, well, the complete peptide mixture that you're analyzing, and you wouldn't be able to see the picture of the whole protein anymore without disturbances due to the enrichment. Will it still be possible to do the analysis like this?
Yes, so I will get to this maybe in a few slides. So we actually did perform a targeted extraction of phosphopeptides in our data. So maybe I postpone the answering of this question to in a few slides.
Thank you.
I will probably get to the rest of the questions a bit later, just to get through the main points first, and then we see how much is still open in terms of questions. So just very briefly, I think I don't want to spend too much time on this, but we benchmarked the COF strategy based on an in silico generated ground truth data set. Basically, the reason why we had to generate something in silico is because there is no good ground truth data set of where we know exactly what proteoforms exist. But in our in silico benchmark set, we could see that we get a reasonable true positive rate at low false positive rates. But what is even more important is that we actually have a working scoring model with well-calibrated and overall conservative FDR models, where you can see the adjusted p-value that COPF estimates against the empirical FDR. You see that we are generally following the diagonal and are slightly on the conservative side, just to show you that the overall method should be sound. So with this, I'd just like to do a very brief disclaimer, because there's a very distinct difference between proteoforms that can be detected in top-down proteomics, because in top-down we can actually fully characterize a protein with unique amino acid sequence and localized PTMs. In bottom-up proteomics and the COPF approach that I'm presenting here, we can only look at proteoform groups, and these are sets of covariant peptides in our data that are differentially abundant across our samples. But the key here is the differentially abundant across our samples, because what this means is that, per definition, all of the proteoform groups that we detect are kind of biologically interesting, because they have to be differentially regulated in the data set that we're looking at. And this is, of course, very interesting, because we get just a few way fewer hits than with top-down, but the ones we get are potentially of high interest. So with this, I now like to get back to the sex-SWOT-MS data, because our idea was not actually to do this on a multi-condition data set, but to look at proteoforms and how they might be assembly specific. So the overall idea here is that the different sec fractions basically work similar to having a lot of different conditions. And if we look at peptide intensities across the sec fractions, then we might figure out whether proteoforms are specifically integrating into a complex or not. So here in this toy example, I again show all the peptides as lines, and they are all peptides from the same protein. And what you see, that there are two orange peptides that are only present in the low molecular weight peak. And this might be very interesting, because then we could say that these two peptides are actually unique to the monomeric form of the protein, and they are absent in the complex bound form. So we applied this strategy of using the COPF approach for sex-SWOT-MS data by using the data set that I briefly introduced before, where we looked at sex-SWOT-MS data of interphase and mitosis cells. And here again, I can say that we could detect both proteoforms that would be due to a difference in assembly state, or they could be, as here, actually a difference in assembly and in condition, or like cell cycle specific. So applying the COPF strategy to the cell cycle data set, we could actually detect 317 significant proteins. So these are proteins that were called by COPF to have multiple different proteoforms. And this was at a 10% FDR and a proteoform score cutoff of 0.1. And what you can see is when we performed an enrichment analysis of the UniProt keywords, then phosphoprotein and alternative splicing came up. So this makes sense in context of finding proteoforms. And also the other terms were largely related to cell cycle regulation, which makes a lot of sense in the biological context. When we further looked at the classification of those proteins, we could assign roughly 100 proteins to be assembly specific, so they differed across the sec dimension, and like around 120 or so were cell cycle specific, and they were actually differentially abundant across the two cell cycle stages. So now I would like to show you some examples of the data. So one is like just a proof of concept example of the nuclear pore complex protein NUP9896. And this is a very interesting one because it actually is coming from a single gene and single mRNA, NUP98, and the precursor protein is autoproteolytically cleaved in the middle to result in two proteoforms, NUP98 and NUP96, which basically operate as completely separate proteins. If we look at the peptide profiles of all the peptides that map to this gene, you will see that you get very distinct sets. So here, all of the lines correspond to peptides that map to this gene, and the colors of the peptides are determined by the groups that Kopf assigned. So what you can see that the yellow and the blue proteoform groups actually have very distinct elution patterns along the sec profile. If we now want to see whether these two proteoform groups determined by Kopf actually correspond to these two proteoforms, we mapped all of the peptides to the protein sequence, and you can see that all of the blue peptides map to the N-terminal region of the protein, whereas all of the orange peptides map to the C-terminal region. And the autoproteolytic cleavage site actually lies exactly in the middle. So this was for us a very nice proof of concept. And what you could actually see is if we do the complex analysis in this data, you will see that the orange proteoform, so the NUP96, is actually integrating in the NUP107-160 subcomplex, whereas the NUP98 protein is not contributing to this complex, which is something that has been known before, but we could do this in a completely data-driven way in our data. So the second example I'd like to show is about an example of proteolytic processing of the proteasome subunit beta type 7 and during proteasome assembly. So what is happening during proteasome assembly? You first assemble all the alpha subunits to a ring. You then assemble the beta subunits on top, and one of the last subunits that enters is the beta 7 subunit. So once the subunit is integrated, it actually needs to be proteolytically cleaved for the full 20S core proteasome to assemble. And when we look at the beta 7 protein and all of its peptides in our data, you see that all of the peptides follow the same pattern, apart from two peptides that Kopf segregated as a separate proteoform group. And these two peptides only contribute to this low molecular weight signal over here, which you can see magnified in log scale over here. So what we know is, as I showed already in previous slides, this high molecular weight signal corresponds to the full 26S proteasome. This is the 20S core proteasome, and this is actually an assembly intermediate over here. So when we looked at the peptide location on the protein sequence, we could see that the two blue peptides actually mapped to the N-terminal region, and the second blue peptide was spanning the known proteolytic cleavage site where the beta subunits propeptide is cleaved off for the assembly to take place. So what we then wondered is whether we can detect this non-tryptic peptide maybe in our actual 20S proteasome. So what we did is we actually went to Skyline and leveraged this like the beautiful feature of DIA that we can go back and re-extract information that we didn't think of before. So we took the non-tryptic peptide spanning this propeptide cleavage site and performed a targeted extraction. And you can see that we've detected a very nice peak in this is now C18 retention time dimension, but if we look at the different fractions here just shown in the skyline representation as different samples, you see a nice elution peak in both interphase and mitosis. And if we aggregate the signal, we actually see that this non-tryptic peptide nicely elutes together with the rest of the 20S core proteasome. So one thing that I would also like to get back to is something that was already asked in the questions, but that already came up in our analysis here, is that phosphoproteins came up as a main term in that was enriched in our proteins that we call to have multiple proteoforms. And and the idea here, or our question, was whether we can detect these phosphopeptides in our data. And our idea was that we could use evidence by absence. So what you could imagine is that if you have a protein and you can see all of its peptides in one condition, and then there is a phosphorylation happening, then the non-modified peptides would actually disappear. So here you see the two orange peptides are highly abundant, but upon phosphorylation of both of these peptides, they might disappear from your analysis. So we thought, okay, this might be a way, even though we didn't do a phospho analysis itself, to figure out such cases. And indeed, we found several very interesting cases. So here is an example of the ataxin 2-like protein, and you can see again all of the peptides for this protein here in these two plots. And you'll see that there were 4 peptides indicated in blue that are highly abundant in interphase, but they are actually decreasing significantly in abundance in mitosis, whereas all of the other orange proteins actually increase in abundance during mitosis. So now we mapped all of the peptides again against the protein sequence, and we saw that they actually distributed widely across the sequence. But then our idea was maybe these are phosphopeptides. So what we did is take a data set, an external data set that was investigating phosphorylation in the cell cycle. And we figured out that there were actually 5 regulated phosphosites that are cell cycle specific. So that I indicated them as kind of these lollipops here on the sequence. And what we saw is that 4 of these known cell cycle regulated phosphosites are actually directly mapping onto our 4 peptides of the second proteoform group. So now we were wondering, if we go back into Skyline, if we could actually extract these phosphopeptides if we look for them. So here, just show one of the phosphopeptide examples. So we did a targeted extraction, and we were super happy to see that we could actually detect a very nice signal for this phosphopeptide in our data. And here again, you can see that there is a nice elution of this peptide across the samples in mitosis. And this is actually in 10 to the 6 range here. And if we look at the interface, we don't really see any elution, like large peak. So this is again sec dimension now, and we are at peak areas in 10 to the 3. So with this, I can conclude that we could actually detect 3 of the 4 phosphopeptides that we hypothesized. And for 2 of them, we even get a very beautiful coelution signal with the other orange peptides here shown in brown and in red. So with this, I'm almost done. I just want to briefly give a quick outlook that COPF is not only applicable to co-fractionation MS data, but it can also be used across different conditions. So we applied it to different tissues of the BXD mouse panel. And so here we measured 8 BXD mice and took 5 different tissues and looked at whether we can find any tissue-specific proteoforms. And overall in this data, we could see 63 significant proteoforms, proteins with proteoforms, and almost all of them we could confirm to be tissue-specific based on a condition-aware ANOVA analysis. So I just want to show one quick example where of the cipher protein, and here again you see all of the peptides of the proteins and the two proteoforms that Kopf detected in orange and blue. And what you can see is that, first of all, the protein is very muscle specific. It's only expressed in heart and quadriceps tissue, and the orange peptides are only observed in the heart. If we now map these peptides back to the protein sequence, we could see they actually span a known alternative sequence region. So what we did is, again, look at what is the alternative splice variant of the protein in this region. And we performed a targeted extraction in Skyline and could actually detect peptides in this alternative sequence region. And these peptides were actually then skeletal muscle specific. So what we could do is basically figure out, based on our data, that there are two distinct isoforms of the cipher protein that are heart or skeletal muscle specific, something that has already been known before, but that we again could see in a completely data-driven way from our data. So with this, I get to the summary. Basically, I hope I could show you that correlation-based proteoform analysis by Kopf can use covariation patterns to assign proteoform groups in bottom-up proteomics data, and it can detect proteoforms derived by alternative splicing, proteolytic cleavage, or indirectly even by PTMs. And in combination with sexwothermos, this actually allows for the first time to systematically assess relationships between proteoform groups and assembly and complex assembly. And with this, I get to the main take-home messages from my presentation. I introduced you to the general concepts of targeted data analysis and how they can be transformed from proteomics to interactomics. And I hope I could show you that sexwothermos is a very great strategy in combination with complex-centric analysis to get consistent detection and quantification of complexes, even across different conditions. And finally, I showed you the correlation-based Kopf analysis, which can allow you to look at proteoform groups in bottom-up proteomics and investigate proteoform-specific assembly characteristics. So just for you to know what you can do with this, so all of what I just talked about is available as R package in the CC profiler framework. So also all the Kopf analysis steps are available in CC profiler, and you can find it on GitHub. And there's not only the package itself, but there's also a complete walkthrough the general steps of the standard complex-centric analysis and even of the DIA analysis with OpenSWOT. And you can find more details in this protocol paper. And for the Kopf analysis, I've also set up a Git repository, which you can find on the bioRxiv paper. So with this, I'd like to thank all of you for your attention and for the many questions, which I hopefully get to now. But I would also like to thank everyone that was involved in these studies that I presented, especially Rudy and my colleague, former colleague Moritz Heusel, who, as I said, generated most of this data, and the other collaborators on all of these projects. And yeah, now I try to get to the questions.
Thank you so much, Isa.
Okay, can you quantify a single protein in different fractions or complexes? Usually one would expect different overall protein expression in different protein complexes. Okay, yeah, so we can detect a single protein, definitely can detect a single protein in multiple different complexes. This is basically what the entire strategy of this proteoform specific assembly boils down to, that we have the opportunity to actually see that proteins elute multiple times in the sec dimension. And we observed this for more than like around 30% or so of the proteins that actually have multiple elution peaks along the sec dimension. Then question by Raja: do you identify occupancies without enrichment step for phosphopeptide occupancy? Yeah. So yeah, as I think I could show you that we could see phosphopeptides also without the enrichment step in our data, but we actually did perform also a systematic phosphosite analysis, but this was not so spectacular. But overall, this works in the data, especially if you have enough fractions, then you can actually extract quite some PTM information from the data, as you could see also with the targeted skyline extraction, where then you would hope to find both the modified and also the unmodified peptide. Another question is, how do you process such large number of samples, all fractions together, all replicates together? Yeah, this is a good question. So I don't know what you mean with process. So in terms of the wet lab protocol, we actually do one sec experiment at a time, but each sample is handled separately. We now have like a 96 wave plate format where we can actually do this in parallel. For the data analysis, I actually, for the Kopf analysis, use a combined information from multiple replicates to get this information. So I actually do an aggregation step before I call the proteoforms. Yeah, so this is how we do this. Then one question, can Kopf take any relative quantitative data, such as TMT? I would actually have to try. So in general, it should work. So if you have, for example, 0,1 normalized data, I don't see why it shouldn't work. So the general idea is that you look at the correlation, and this should be agnostic to the absolute values. So if you didn't mess around with the ratios between samples, this should be fine. But I haven't tried it, and I'm not a TMT expert. Then one other question, how does the sex-swath approach compare to genomics-driven proteoform discovery? I guess the sex-swath filters for functional differences of proteoforms. Yeah, so I guess with the genomics-driven approaches, you're referring to RNA sequencing approaches for alternative splicing, for example. And what we saw is that we can, of course, only see a fraction of what can be discovered by genomic approaches. So, for example, you can detect hundreds or like thousands of alternative splice variants, which we cannot detect by the Kopf approach. But as you also correctly said, what we can detect are actually functionally relevant proteoforms that show a difference between conditions. So we are blind to any kind of proteoforms that are just present in the cell but don't have an impact, but we could see the ones that are actually different across our data set. Yeah, one question, does it have to be SWAT-MS for DIA? No, definitely not. So you can use any DIA setup that you would like to. So this is completely platform independent. And could you quantify the phosphorylation in protein complex? Yes, so I mean, in general, we could, I mean, this is what I just showed in the slides. So this is, so I actually don't know which complex this corresponds to, but what I know is that this is not the monomeric form of the protein. So we could see that this protein elutes in a higher molecular weight than you would expect for the monomer, but we don't know exactly for this example what the interaction partners are. But in general, you can see the complex information for these phosphopeptides in our data. Okay, then one other question, can Kopf be used to analyze regular DIA data, not the co-fractionation? Yes, so this is exactly, I think I answered in the last data with the mouse tissues. But what is important here is to say that we really need a lot of variation. So in this data set, we have a very diverse set of conditions, and we expect almost all of our proteins to change in abundance. And this is kind of very crucial to the strategy, because this correlation-based approach only works if you have differences. And if you don't have any differences, you will get non-relevant correlation information. And then what was the reproducibility of your peptide detections along experiments? So I assume that you're talking about proteoform detection. So this is actually fairly high. So as I said, I did this actually for the different replicates combined, but I also performed initial analyses on each of our replicates and conditions separately. And for the assembly-specific proteoforms that were non-condition specific, we could see a very high reproducibility of the calls across the different samples. Then have you tried with IEC with SWOT MS, and if so, is there a significant difference between SEC methodologies? I haven't personally tried, so there are a lot of different strategies that you could use. I know that other people in the Ebersold lab have also used Blue native page separations, and this worked extremely well for other sets of complexes that would, for example, also be larger and potentially membrane associated. I have not tried it with IEX, but from the methodology, like from the data analysis point of view, it works for all kinds of separation techniques. Although the idea of using the molecular weight as an additional factor only works for separation techniques that actually involve the molecular weight of the complexes. Okay, and I think there's one final question. Can you use the SEC-SWOT DRA coupled with other bottom-up or top-down MS to determine the stoichiometry within selected protein complexes? And can you convert the abundance factors from mass spec analysis for one selected protein into copy numbers in the complex? Okay, so in terms of stoichiometry, this is actually a very cool question, and we have looked at this in context of the septin complex, which is present both as like in multiple forms, and they have multiple exchangeable subunits. And what we could nicely see is that the exchangeable subunits are at a much lower relative abundance compared to the constitutive subunits. And we could even calculate these stoichiometries, but unfortunately our data is maybe a little bit too sparse to get like a full-blown picture of how well this works. So I would say, so CC Profiler actually reports stoichiometries, but you should look at them with caution, I would say. In terms of copy numbers, I haven't done it, but in theory, it could be possible if you calibrate it right. And another question, in complex protein discovery, can you summarize the weak points of SEC-SWOT and the bioinformatic workflow exposed here? So the weak points, I would say, are that you definitely need prior information, as I said. So this means that if you use a non-model organism, for example, you're depending on information that might be from homologous mapping or things like this, and you have just very little information. And then I would say that the complex-centric analysis workflow doesn't necessarily work very well. The other thing is that you need quite some material to perform, like from the experimental side, to perform like a full-blown SEC-SWOT MS experiment. And I mean, I think it works already fairly nicely, and we could do it with tissue samples, for example, as well. But overall, you need quite some amount. So it's still some way to go, for example, to get to clinical samples. And one maybe other point from the bioinformatics side, what did I want to say? The weak points is that so this was now the weak points of the bioinformatics workflow, is that you need a sufficient resolution to actually be able to derive signals with this. So if you, for example, have noisy signals, this gets very, very challenging, and you will need sufficiently high quality data for the analysis to overall work. And of course, Seg-Swarth-MS in itself is a strategy for kind of stable complexes. So if you want to, for example, look at transient differences, like very small differences between perturbations where you have small, like some minor rearrangements in complexes that are not super stable, I would not recommend to use Seg-Swarth-MS because you will see the rearrangements on the big scale. So if you have a massive rearrangement of complexes, as for example in the cell cycle or during differentiation processes, then this is a great strategy to use. But if you're more interested in like minor perturbations, I would recommend something else.
Well, Isa, thank you so very much.